Hello everyone, I hope you're well. Today we have another webinar related to spaces. And before we start, I would like to just remind you the major rules for the webinar. And I hope you can hear me well. If you do hear me, please type yeah, okay, yeah, I see. Yeah, so we can hear you. You can hear me. So just a few rules about the webinar. You will be muted during the webinar, and if you have any questions, please type them in to the questions box that you can see in the go-to menu. You will be prompted to follow up us and find out more about webinars or upcoming webinars, either through LinkedIn or other social networks. Once a webinar is finished, you will receive the recording of the webinar that is freely available through our YouTube channel. Okay, so as I said, today we have another webinar that related to the spaces, and before we go to our today's presenters, I'm gonna just remind you why spaces and the tools to explore them are important. Yeah, as we all know, the chemical space is tremendous and it's huge. But in many cases, or in most cases, we need not the molecules, but the samples. And if you have looked through the available chemicals, we're not even reaching to that number of the all possible molecules. We're still in millions of the in-stock items, and obviously trillions of make-on-demand compounds. And those trillions of compounds can be synthesized to you by a few companies. And in most cases, the molecules are delivered with a high success rate. The reason of this is that the spaces are created not based on some intuition, but based on the chemical knowledge. And we have few examples of those. What we're doing at Chemspace is trying to link the knowledge of heat discovery with a knowledge of procuring compounds through different sources and from different databases and chemical spaces. And today we have two speakers, and that's really great pleasure for me to introduce to you Dr. Marc Nicklaus. Is the head of computer-aided drug design group at Center for Cancer Research and NCA NIH, and he will present the approach to creation of ultra-large libraries of compounds that can be synthesized and indeed delivered and tested. With that, I would like to pass word to Marc and make him a presenter. Marc?
Okay. Okay. Can you see my screen?
Not yet.
Not yet. Let me. Okay. How about now?
Yes.
Okay. Move this out of the way. Okay. Yurii, thank you very much for the invitation to talk about our SAVI project and SAVI database. And I said, I say our intentionally because Yurii is actually a part of it and co-author of our paper about SAVI. So as Yurii said, the space of synthesizable molecules is nearly infinite. And he said 10 to the 63, and many people say 10 to the 60. And if you compare that with drugs and candidates in the development there, I mean, we're talking thousands of molecules. And the question is how much undiscovered chemistry is in there. And, you know, this number, okay, 10 to the 60, people say, yeah, I've heard it. What does it mean really? So think about the number of known molecules. And I said 100 billion, you know, for real space. And, you know, it's a trillion. It doesn't really matter. This is, if we say this is a grain of sugar, okay, then what is the remainder? And if you ask that to many people, they say, well, you know, okay, I don't know, like the size of a city or the size of Earth. No, it's not that. It's the size of the entire Milky Way galaxy. This is the druggable space that we don't know versus a grain of sugar, okay? So now what you want to have indeed are synthesizable molecules. Now, what is easily synthesizable? Let's say 1 out of 100,000 compounds is easy to synthesize. If we look at that versus the galaxy, we still have 1 million stars to look at versus 1 grain of sugar that we have looked at so far. So the task is, you know, enumerate these molecules, or at least a chunk of it, right? And then apply computer-aided drug design techniques to it, docking, pharmacophoricity, and you're done with drug design. Well, it's not that simple, but at least it's a useful step to take. So, SAVI, the synthetically accessible virtual inventory. What's needed? What components need to go into it? Okay, first, you need building blocks because you don't want to start synthesizing those, then it becomes complicated and lengthy, okay? And here we have to, again, thank Yurii, who made the Enamine building blocks available to us over the years in various iterations. The second part is you need some rules to put these building blocks together. And here we use something that is a little bit different from what other similar efforts use. We use transforms that come from the LHASA project, and you will see a slide just in a minute. And this is has mostly been done by Phil Judson, and Phil is actually in the audience, so I say hi, Phil. Okay, and finally you need a toolkit, some chemoinformatics method to put these building blocks together with the transforms to novel compounds. And this has been done or developed by Wolf-Dietrich Ihlenfeld in Germany and his Cactus chemoinformatics toolkit from his one-person company, Chemistry. And we in my group, we have, you know, run these jobs to generate actually the SAVI database. Okay, building blocks. So why did we choose Enamine building blocks? There are other building blocks out there, as many of you know, okay, but the crucial reason was that is Enamine actually synthesizes the molecules? You can generate all kinds of molecules in the computer, and then what? If people don't synthesize it for you, you need to have a lot of synthetic chemists, and NCI doesn't have a lot of synthetic chemists. So we need someone to synthesize them for us in general. The subset we used for what I will describe to you, which is the SAVI version that was generated and made available in 2020, was about 152 building blocks. And the cutoff was 1 gram of it should be available in the Enamine repository. And it's important to point out that these are essentially, or were then, the same building blocks used for the Enamine real database, okay? And if you check which ones of these building blocks match at least one of the transforms, there were about 143 out of the 152,000 that matched. So this is what we used. Now, these transforms, let me spend a minute on it. This is essentially a revival of the LHASA technology. That was the computational embodiment of the E.J. Corey's retrosynthetic Nobel Prize winning work. Very importantly, it's an expert system type approach. It's not machine learning on large data sets. It consists of a language pair called CHMTRN and PATRAN. And it goes really way beyond the SMERCS transforms that are widely used otherwise. Now, the LHASA project and the LHASA code that existed, and it most likely doesn't exist anymore, or it doesn't run certainly anywhere, was retrosynthetic. SAVI conceptually is forward synthetic, so we make new molecules. The parser and the execution engine were developed for Cactus. Very importantly, this LHASA approach with a more detailed description of the chemistry logic allows a coring system, add and subtract statements that say this will work better in the hood or this will work less well. And crucially, and maybe the single most important word in our project is the kill statement. When the logic says, don't even try it, this will probably not work. Okay, and the scoring also allows you to focus then on most reliable synthetic predictions. So we got access to the regional knowledge base of the LHASA project, and that's about 2,300 transforms. And we started using a few of them, but it became clear right away that a lot of modern chemistry, such as Suzuki coupling, was not in this approach because it petered out in the 1990s. So we had Philip write new transforms for SAVI 2020. We added about 40 new transforms. Most of them are coupling reactions. We have some ring forming, a subset. And as I said, the number of transforms used for SAVI 2020 was 53. Okay, just an example. This is a cutout of one of these transforms. And you see it is quite different. If you know SMERCS, and many of you may know SMERCS, this looks different. There's a whole bunch of text that basically describes to a chemist, what's going on, what can you do, what can you not do. And of course, this is read by a computer, okay? So that's why we say it's smarter than smarts. So you have statements subtract 10 and subtract more. So you have an amount of subtraction or addition of points, so to speak. And at the very bottom, you see kill if withdrawing bond on atom number 6 is off path. And then this proposed reaction is thrown out right away. Okay, without going in detail through the transforms we've used, on the left side, these were the transforms from the old knowledge base, and they all have a 4-digit ID number. And again, feel free to quickly look at it. And then on the right are the newly written transforms that we used for SAVI 2020. Now, I said that the SAVI project is conceptually forward synthetic, but for a bunch of reasons, we, again, mostly Philip, wrote the transforms, or we decided to continue writing the transforms in the retrosynthetic way so that they could be used for retrosynthesis analysis. So that means you have an interesting workflow. You have the building blocks, and then you do the matching of is a given structure in the building block set matching with reactant 1 and reactant 2. You make a reactant pair, and then you ask the transform based on the product generated from this reactant pair. Can you propose a synthesis? Right? And if this LHASA reaction procedure says, no, I cannot make a synthesis, there's all kinds of things that prevent it, then the reaction obviously is killed right away. You skip the product. And then in a second step, the question is asked, are the reactants that are proposed by that LHASA transform application, are they the same as the input? Because you can have cases where that's not the case. And if that's not the case, then you say, well, this is not what we wanted, so we ignore this reaction. We want to have the reaction exactly based on A plus B gives C, the way we wrote it down and tested it here. Okay, and you see that here. Very importantly, all of SAVI 2020 are single-step reactions. As I just said, A plus B gives C. Okay, the simplest chemistry that you can imagine. The initial reactant pair number that we looked at is about 3.6 billion. And for a little bit more than half, the reaction logic actually killed it and said, no, don't do it. Okay, so we were left with 1.75 billion reactions that were accepted by the reaction logic. This scoring that I mentioned allows us to categorize or classify the products into different classes, quality classes, if you will. And the plus class at the top are reactions that did not encounter any of the detractions, any of the subtract statements. Okay, and then you have, or we defined classes where increasingly severe subtract events were encountered in the logic. Okay. What you also can see is that the number of unique structures is a little bit lower, and that is not undesirable. That means that you have, for about 10% of the SAVI products, multiple possible synthetic routes. And this is, again, actually a welcome feature. This is not a flaw in the approach. So we published this paper about this set, and we have made in 2020 the entire set available, downloadable from this link. Okay, now this is pretty large. If you download it in SDF, it's more than 4 terabytes of space, and if we also have it as a SMILES table, which is obviously more compact, then it's still over 1 terabyte. So this is what you currently can obtain for completely freely, and do with it whatever you want to do. Now, so we have more than 1 billion molecules. Well, are they good molecules? Are they interesting for drug design? So we tested the SAVI 2020 set along all kinds of properties that are typically looked at for drug and drug-like molecules, and I'm just showing a few. More are published in that paper that I just showed. The total polar surface area, the non-flatness, the fraction of sp3 atoms, and then the drug likeness score, this QED score. And if you compare that with the original drawing in the original paper, you see that actually it looks darn nice. So all the properties that we looked at look good. So that told us, okay, these molecules are reasonable. They are drug-like looking. Another nice built-in feature is that you automatically have SAR series into the SAVI approach. Not just the SAVI approach, a lot of these ultra-large spaces have that property. But if you think you have essentially a matrix, in this case, we have 143,000 actually matching molecules. And so there are actually only a few cells are filled. And in reality, it's a 3-dimensional matrix because we have 53 transforms, but you can even project it on 2 dimensions. It's easier to visualize, and the math is not that wrong. So if you do the math, you will see that for each molecule, like the one in the center here, you have on average 12,000 analogs. So this is cool, right? So let's say you find a molecule that's active, okay, and this is just one example here. This is the click chemistry that we implemented, and you say, okay, this is reactant A and reactant B gave that molecule. What are analogs of it? And so you keep reactant A constant, and you look at all the reactant B's that were found and that were successfully coupled in the computer, and you have 700 molecules here. And you can then do the same thing the other way around. You can keep reactant B constant. And so this little example already gives you about 1,500 analogs to look at, okay? And they're not just methyl-ethyl kind of changes typically, because we said we want to really couple blocks together. We have not in the set of transforms used functional group interchanges, which you also can, but we haven't done that. So you have larger changes, and this may be good and bad depending on what your drug development project is. Okay, so we calculated the overlap of SAVI with other databases, and at the top line you see the overlap with the real version back then in 2020, which had about 1.2 billion molecules. And the overlap was about 8%, and that's good. That's a very good result, because remember, it uses essentially the same building blocks, or it used the same building blocks as real. So you expect that to happen. And it also shows you that we're generating reasonable molecules and not something bizarre, because the assumption is that Enamine with real has generated reasonable molecules, not bizarre stuff, right? At the same time, more than 90% is not in real. Okay, so there's a lot of novelty in the SAVI set. Okay, and we compared it with the IRL from a ChemNavigator, which was then purchased by Sigma and which merged with American Darmstadt. So, and there the overlap is much, much smaller. Okay, of course, the size of the molecule, the size of this database is also smaller. The overlap with PubChem is 0.3%, but it's still 5 million molecules. So, you know, I mean, we have resynthesized in a single step 5 million of the PubChem molecules. And interestingly enough, we even resynthesized some of the building blocks we used, which is also not too surprising, because I'm sure Enamine uses, you know, existing molecules to synthesize more building blocks, so that makes sense. Okay, one analysis was what ring systems have we created, and these were discussions that also were helped by Peter Ertl, who was in the audience. So hi, Peter. And this is a little bit difficult to determine, because ideally you need a set of all the existing ring systems. And people would say, well, you know, maybe CAS registry is the best source. Well, you can get access to it. So we estimate that we have maybe generated 1,000 or more than 1,000 novel ring systems. So this is also interesting, right? You say we have made rings that have not been seen before. So, okay, so we have a lot of molecules. We have molecules that seem to have the right property. Can they be synthesized? Now, this is what Yurii pointed out at the beginning. So far, we have, or we know of, about 170 SAVI 2020 products that were synthesized in the context of NIH drug development projects so far. And this was typically anti-cancer, but also anti-inflammatory and antiviral projects. And they were all from the plus subset, because that's what we usually look at in silico screening. Some were synthesized by NCADs at NIH, you know, which most of you probably know or know about, and most were synthesized by Enamine. Okay, and the success rate was 97%, which was astounding even to us. You know, I would have been happy with 80%, right? So maybe we have been lucky, but it seems to work quite well, and the transforms in their strictness, you know, to throw out a lot of stuff that's doubtful, seems to have worked quite well so far. Okay, with this, I'm coming to the end here. What is coming for SAVI? What are we working on and looking at? As I already said, you know, for SAVI 2020, we had 53 transforms and we had 153,000 building blocks. For SAVI 2023, or 20X, we have about 120 transforms now. We got from Yurii, you know, an updated building block set a little bit earlier with 238 building blocks. And I estimate the size of this new database would be between 4 and 10 billion. It's a large range because we don't know yet how these new transforms will match with the existing building blocks. So this is kind of in the works, okay? Now, there's also this exciting made building block set from Chemspace, 770 million. I think by now it's already 800 plus million. It grows by 10 million per week or something like that. So Yurii can say more about it. We have some transforms don't have many matching reactants, so we call them starved transforms. And this is certainly something really interesting for us to look at this made building block set for the so-called starved transforms and to fill it up. Okay, if we would use all of the 770 million made molecules, I estimate, this is a wide estimate, you know, that it's more than 10 to the 16 molecules in single step synthesis. We are not yet at 2 or 3 step. Just single step synthesis. So this thing explodes on you, okay? Indeed, trillions, more than trillions. And the challenge is, of course, the big question, the $64 billion question, if you will. How do you pick interesting chunks from these huge spaces, you know, for your target, for your project? And with this, I would like to acknowledge all the people worldwide who have contributed to this project, and I will be happy to answer any questions.
Yeah, thank you, Mark, for this really nice talk. We have a few questions. But the first question is, what does the subtract statement mean?
Well, that's a what does it mean? You know, we have Phil Judson here in the audience. If you really want an expert answer to that question, we should ask him, right? But it's really well, it's scoring. If you have a group somewhere in the molecule that may make the reaction less likely to succeed or at a lower yield, there's many reasons why you can have a subtract statement. You know, this basically says, you know, give the whole reaction a score, default score, and even the default scores are different between transforms. And then you basically, you know, you use the knowledge that chemists say, this will work well, this will not work so well. So this is what the subtract does, incorporate that into a scoring system. Now, there is also a qualitative scoring system that we developed. So again, this is all published, you know, so you can look it up, because the question is truly, you know, what does number mean? Subtract 10 versus subtract 15. And we had a lot of discussions, you know, mostly with Phil, you know, about can this be done in a little bit less quantitative way, which in a way doesn't make any deep sense, right, to have specific numbers.
Thank you. Another question. Could you think for the future also about two-step reactions? Would it be computationally manageable at all?
Well, that's a very good question. Thinking, yes, computationally manageable, question mark. You know, we estimated for SAVI 2020, if we reuse all the products, right, the 1.7 or even just the 1 billion, right, as building blocks, plus an existing building block. So it's 2 steps, not 3 steps yet, okay? That we are already above 1 trillion. So no one really enumerates 1 trillion molecules, right? And then you have to think again, you know, how do I pick the interesting chunks? And this is, of course, what we want to do and what everyone wants to do in that field, right? When you have these 1 trillion plus molecule databases.
Yeah, thank you. And that actually, you know, brings me to another, you know, to another question. So, what do you think the future of the databases? Because as you just said, trillion, it's really, you know, hard to enumerate. So, what do you think we should look into to, you know, to go beyond trillion?
Well, you obviously need to start bringing the target into the whole approach and the you know, evaluation, maybe even scoring and these kind of things. I mean, if usually you say, okay, I have a specific target, a 3D structure that I do docking with, or whatever it is, right? And so you cannot dissociate it anymore, right, from the structures that you're looking at, because you want not 1 trillion, which you cannot handle, and certainly you cannot handle 10 to the 16, 10 to the 20, or 10 to the, you know, 26, like I think, what is it, Merck's Massive, and then the GSK XXL or whatever, right? These huge databases, they're all not enumerated. So how do you find the active, I mean, how do you find the interesting stuff that gives you active molecules in an efficient way, right? And that's, as I said, the $64 billion question. And I don't have a good answer, but we're discussing it, and you know, I'm sure you guys discuss it too. Right?
Yeah, thank you, Mark. That's indeed the $60 billion question, and with that, I think I would like to thank you again for this really great talk, and would like to move forward to our next speaker today. Let me just share my screen. I hope, can you see my screen now? So today our next speaker today sorry, can you see the screen or not?
Yes.
Okay.
Now we can.
Great. So our next speaker today is Dr. Nadya Tarasova. She's the head of Synthetic Biologics and Drug Discovery Group at the Center for Cancer Research at the NCI NIH, and she's really great expert in exploring such huge chemical spaces and chemical databases like SAVI. So with that, I would like to pass the word to Nadya, and looking forward to the presentation.
Yurii.
Just sorry. Yeah. Okay.
Looks good.
Looks good. Can you see the screen?
Yep.
Good. So first of all, thank you. Lots of thanks to Chemspace for the invitation to present here, and thank you all for tuning in. We all know that drug discovery is a fully slow process. The efficacy of global drug discovery can be measured with the number of annual FDA approvals, because FDA evaluates drugs discovered anywhere in the world. And this graph shows that after a prolonged decline, almost 2-decade decline, that was caused probably mainly the overestimation of the power of high throughput screens. The number of approved small molecule drugs is finally started to increase steadily. There were some hiccups caused by administrative measures and COVID, but overall trend is very positive.
Excuse me, Nadya, I think we're still on the first slide. At least it's what I see.
On my advancing. Okay. Okay. So this is the graph that I meant to show. So this is the decline, and this is what's happening right now. So we see the increase, and it is particularly noticeable when we talk about anti-cancer drugs that have been on accelerated discovery slopes for the past decade, both for biologics and for small molecules. And even the acceleration correlated very well with the increase in the number of drugs discovered with the use of protein structure-based approaches, suggesting that modern computational methods have a huge impact on drug discovery. Virtual screens became a primary approach in drug discovery for several reasons. First of all, the accuracy of docking software and algorithms evaluating binding energy have improved significantly. Also, sufficient computational resources, and we need a lot, like hardware and cloud computing, have become available. Also, significant advances in protein structure determination technologies have been made, and we need structures. And what's hard to overemphasize is that large virtual libraries of synthesizable compounds became available. However, many challenges in finding tumors still remain, and one of the major ones, particularly for cancer, are so-called undruggable targets that lack deep pockets in their structures. For cancer, we have to deal a lot of protein-protein interaction, and that's both for the drugs that exert their action by impacting tumor cell growth and for immunotherapy development. And we all know that immune therapy are the only one that are producing curative effects. So there are also lots of oncogenes, intrinsically disordered proteins, and lots of transcription factors. So with the development of super large databases, the question arises, is it really impossible to target cleftless proteins with small molecules, or we have been searching in the wrong part of enumerated chemical universe? Marc and Yurii already alluded to the size of the chemical universe. That is beyond astronomical, well beyond astronomical. And the size itself constitutes a big challenge because 10 to the power of 16 compounds that potentially can be synthesized, right? So generated using SAVI approaches would occupy 100 petabytes of space. Nobody has resources like that. So this is why lots of accessible parts remain as spaces that are presented here as purple circles, in opposed to circles in other colors that actually have been generated as files and can be used in searches. However, mining even this enumerated constitutes a very large challenge in itself. The problem is that docking with calculation of the binding energy still remains the most reliable way of screening of virtual libraries, and it is very slow. The method that we use, this is ICM Pro for MolSoft, takes about 30 seconds per compound. And we're lucky to have pretty good computational resources, but even so, running 4,000 parallel processes on an NIH supercomputer would allow you to screen this 1.7 billion compound library, for instance, in 148 days, and will take over 14 million CPU hours, which is not, of course, practical. So what are the alternatives? So there are at least 3 possible solutions to the slow docking. One is widely used, and this is fragment-based design, which is widely used for a good reason. So it is effective, but the major drawback, so it doesn't explore sufficient number of possible linkers. We use the diversity library design. So, and the third would be, which is not here yet, but in some parts it's coming, and it's right around the corner. This is faster docking using GPUs, and we also should be thinking about quantum computers, although they are not here yet, but they may arrive quicker than we all think. But nevertheless, meanwhile, we're using the diversity library. So the approach is consists of first generating a highly diverse acceptor full library. So 15 million compounds, which we can dock more or less comfortably. So then we use the produced hits to search entire database for similar compounds. And this could be, so these are ligand-based searches that could be 2D searches or 3 searches. And then, we usually get about several million hits that we redock. So and just, to kind of a little bit ahead of myself, so the answer to the question is partially yes. So a virtual screen indeed reduces the number of non-druggable targets significantly. So these are just some examples of classical undruggable targets for which we have been able to identify and confirm ligands using this approach. So how do we do that? So here is the typical workflow. We usually start from verification of X-ray structure suitability. And for that, we run a test docking screen of Chemspaces in stock compounds because they're reliable, available, and they're of sufficient size. So they have been working really well for us. So then we test the hits. So we do docking really carefully. So, and that's why the average number of active molecules across many targets that we investigated is in the range between 30 and 50% of all tested compounds, which is well above average for the field. However, we also noticed that the overall pattern is all or nothing behavior, which means that some X-ray structures yield close to 100% actives, whereas others had practically no activities. So, a good example is KRAS. So it is a tough target, but it was fun to work with it, particularly because there are over almost 300 structures of it in PDB. So, and we were able to identify some that were suitable for virtual screens, while some, in spite of being very high quality crystallographically, having high resolution, produced 0 hits, no matter what kind of methodology screening we use. So after we do the first test, and if the structure proves to be suitable, then we run docking of SAVI diversity set of 5 million compounds. Because it is more diverse than Chemspaces. However, the compounds need to be synthesized. So we synthesize and test those hits, then we combine them with Chemspaces hit and run fragment-based or 2D similarity search of entire database for analogs. And for 2D searches, we use either MolSoft's Giga or Arthur from NextMove, or very successfully have been using FiveWalker from OntoChem that is based on Google's BigQuery. So this is probably the fastest and the most user-friendly approach. However, you have to pay big bucks to Google for the use of BigQuery. And in addition, we found that 3D-based similarity searches produce much better results. So we have been using so-called atomic force field, atomic property field, which is a machine learning technology that can be used not just for screening, but also for scaffold hopping. It implies superimposition of known hits and generation of 3D pharmacophore potential that consists of hydrophobic, aromatic, proton donors and accepted charges, and so on and so forth. The generated pharmacophore can then be superimposed with every molecule in the database. Because many have rotatable bonds and can exist in multiple conformations, you have to superimpose to every conformation. And that's why EPF actually takes almost as much time as docking. So that is why MolSoft has developed a new faster version of it, so-called Rapid SSTR Discovery Engine, which is faster for several reasons. First of all, it uses pre-generated databases of conformers. It can also run on both CPUs and GPUs, and of course GPUs are much faster, about 30 times. Generation of conformers requires significant computational resources. However, we have to do it only once. And using NIH supercomputer cluster, we have now generated conformers for SAVI 2020 real, the nearest real about 6 billion, Wuxi Galaxy 1.7 billion, Enamine made the older version 600 million, and older version of Chemspace Freedom, that is 0.16 billion, but also the newer version of Chemspace Freedom, which is 5 billion in the works. So in total, currently, conformer database is about 10 billion compound, and it can be downloaded from Allsoft server. Although you can also write to me and I'll provide you with the link. But keep in mind the size, which is 5 terabytes for real, 1 terabyte for SAVI, and 2.6 terabytes for Wuxi. So after using and identifying similar compounds, we dock all identified ligands, and then use structure optimization using traditional medicinal chemistry. And this way, we were able to identify compounds with binding affinity in at least micromolar and frequently nanomolar activity for most of the targets, even very challenging ones. An example of the discovery scheme application for STAT3 N-domain has been published, so that's the reference. The domain is a very important target, not only for cancer, but also infectious diseases. But it is a challenging target because the only cleft it has is very shallow, and drug-like density index for it is actually negative. Nevertheless, identified inhibitors are in preclinical development already. The approach with the use of diverse libraries is effective. It's not without limitations. The bottlenecks here is the diversity set and the libraries themselves. Paraphrasing Julia Child, there are 3 most important properties of the libraries. These are diversity, and diversity, and diversity. Library diversity can be increased by diversifying the blocks, but also the increasing the number of different chemistries are used for generation. And Marc and Yurii already alluded this question, because there was lots of focus on the blocks, but what are the optimum chemistries for generation of drug-like molecules? So in order to at least, peek into this question, we have docked SAVI diversity set into 39 well-characterized pockets, analyzed the number of hits produced by each of the transform. So Robert Song, who is also present here, has conducted all those searches and docking, which of course, took significant amount of computer time. And we have investigated 2 types of pockets, small molecule pockets and protein-protein interaction type interfaces. So all the structures that we use have been verified for their suitabilities, and by redocking existing ligands or known ligands. Many of them are in the clinics already, and it's remarkable that the RMSID was fairly low for all of them. But the most striking result is that, first of all, the drug-like density measure descriptor had a positive correlation with the number of hits, which could be expected, but the correlation was statistically insignificant. And this is important to keep in mind when using these things. And because the concept of DLED has been developed based on PDB data more than a decade ago, and probably need to be updated with the inclusion of modern drugs. And talking about modern drugs, I want you to look at these inhibitors of several targets that previously believed to be undruggable, but now are in the clinics, most of them, although just phase 1 clinics. So, you know, what obviously strikes is the complexity of those molecules, right? The size of those. And look also at log Ps and log S. So most of them would have been thrown during the filtering of the databases that we use. So that's, you know, suggests that the definition of drug-like probably I need some editing, taking into account the modern drug development. So the other striking result that we got was that hit rates for 93 transforms used for SAVI generation differed a lot. So the differences were more than of order of magnitude. So the productivity of some of them were really low, while some were really high, which means that, you know, enumerating those probably doesn't make as much sense as enumerating the others. And the transforms that produced the most hits were CC coupling reactions. That includes Gashiber coupling, Suzuki-Miura cross coupling, and Hiyama carbonyl-active cross coupling. So this is again something to take a note about for the generation of future databases and increasing the number of the blocks. So definitely we need more borates and silanes in order to use these chemistries. And as Marc mentioned, the whole effort, all described the research, was contribution not just my group, and here is the current members, Robert, Remington, Cody, and Kevin, but also it was an international effort. So we have contributed, you know, benefited a lot from participation of lots of NCI investigators. Lots of companies contributed, and of course Marc's group was a dear friend and a huge contributor to all of that. And we're particularly thankful to Ukrainian chemists who, in spite of horrific difficulties, keep reliably supplying us with compounds and make everything that we need. And with that, I would be more than happy to take questions.
Yeah, thank you, Nadya, for this words and this really great talk. So a few questions. Could you please share some secrets on how to reach such a high hit rate in virtual screening?
So this is basically docking and redocking. We do docking in 3 steps. First, we run a quick screen because at least the ICM Pro that we use is based on Monte Carlo approach, which means that it takes every conformation of the molecule, throws it randomly into the active site, and calculates the energy, which implies that there are going to be false negatives and false positives. So there is little we can do about false negatives. So, but in order to get rid of false positives, we do redocking of the hits that are identified on this first step, docking them significantly slower and with many trials. So, and after that, we do manual redocking. So here we can verify the pose, you know, all the interactions, you know, to make sure that there are significant impact of hydrogen bonds, all high energy interactions. So, and that's why, you know, I think we're getting better results than some. And I think it's a responsible way of approaching, because not only the compounds are difficult to make, and chemistry is environmentally unfriendly field of science, but also the assays cost a lot.
Yeah, totally. And when you say that you have this, you're using this, three-step kind of hit selection. What is the number of compounds you have, for example, on the second step of docking, and on the like third step, last step of manual docking?
So that's a good question. So definitely the number of the hits we get on the first step differs a lot depending on the pocket. So, but usually for practical reasons, we select something between 2 and 5,000 hits for the second one. And for manual redocking, it also depends. We redock something in the order of 15, 30 top hits. You can kind of feel when the scores start dropping down sharply. So that's where we end. Actually, it's more manual docking and more work when we get away from diversity sets, because there are lots of compounds that are very similar and they come up with very similar scores. So that's when selection becomes more challenging. But that's, the numbers, typical numbers that we use. And for structure verifications, we don't order a lot of compounds. We cannot simply afford lots of them. So usually that's between 5 and 10.
Thank you. Maybe one more question that is related to the procedure that you described. Have you ever thought on docking the whole SAVI data set, at least the one that is available now?
Trying to dock it. We will be trying soon. Simply, we do not have computational resources to do this, as I mentioned. So with the current computational resources, it would take 148 days, and that would occupy all our nodes, and we won't be able to do anything else. So that's why we haven't done it yet. However, I have mentioned REACH, that's a new docking protocol developed and software developed by Moroz that is still in beta stage, but hopefully will be implemented soon. So, and we plan to do just that, to dock it on GPUs.
Yeah, thank you. And that brings us to another question. Do you think there is a possibility to increase the success rate of virtual screening? And maybe, maybe that's really software dependent?
No, it is hard for me to answer the question about software dependency, because we haven't benchmarked two softwares in parallel. So we did try to do this in the past, but on very few targets. So trying to compare Moroz and Schrödinger. Right, so, but I wouldn't be making any statements because we simply do not have enough data to support any solid conclusion on that. So definitely the improvements are possible, but again, the software, right? So, but the major bottleneck are still the structures of the proteins, because, and that's not crystallographer's fault. So that's the problem, is that crystal packing can impact the structure dramatically. Again, I mentioned KRAS, so where there are almost 300 structures, and of course, if you compare them, the fold is the same. But there are still significant differences, not just in side chains, but in the position of the loops, relative positions of the helices. So obviously, the structures are different and would produce different results. So what we're using a lot currently, and CODI is doing that in our group particularly successfully, is trying, if you know a ligand, So you're trying to optimize the structure about this non-ligand computationally. All right. So, and another, of course, approach, it's classical. So if you have a ligand, you co-crystallize it and use a better structure that has been already adapted. Your pocket, a lot better. So there is also a lot of efforts to use AlphaFold for docking, and we are trying this too, but in combination with molecular dynamic stimulation. And David Bell, who is collaborating with us on this project, is here also. So I don't see any reasons why it shouldn't be working.
Yeah, thank you. And actually, you know, thank you for the answer, Nadya. And actually, you know, the question regarding AlphaFold was on my list. So have you had a lot of experience with docking, you know, into the or against the into AlphaFold generated structures?
I would say that we had lots of experience, and would have been, you know, just serendipity, but we did have one successful try. So it could be an exception. But unfortunately, you know, I'm not at the freedom of name some targets, because these are all collaborative projects, right? So so I'm supposed to keep it private for now. And it was a very difficult target indeed. So, but the results sort of surprised me a lot. So it wasn't the binding is in micromolar range, but at least there is binding. So that's why I'm optimistic.
Yeah, thank you. Thank you for the answer. And they were looking forward into the improving our efforts towards certification of, you know, better hits or more hits was just using AI or ML derived methods. I think, yeah, with that we have a few more questions, but it's just in the sake of time, we'll follow up with you via email. And with that, I would like to conclude and finish today's webinar. And again, I would like to thank you again, both Marc and Nadya, for this really terrific presentations. And we're looking forward to hear more on the development of SAVI, and obviously ways to explore ultra-large chemical spaces from you. And maybe in a year or so we have this updated webinar with your thoughts and hopefully new developments. So thank you again, Marc, Nadya. Thank you everyone who joined us today. Yeah, stay safe and have a good one.
And you say stay safe, and best of luck and everything. Thank you.
Thank you.
Bye.